Assalamualaikum and a very good morning to our respective lecturers and fellow classmates. We are from Group 2, supervised by Professor Madhya Dr. Asma binti Hamid, who would like to present on the topic entitled a NR2F1 agonist, C26 drugs, which is a novel therapeutic approach in preventing metastatic tumor.
Most cancer deaths are caused by metastatic cancer. Metastasis occurs when cancer cells break off from the original tumor, which is also called the primary tumor, travel through the body and begin to grow in other tissues and organs. Metastasis causes death by damaging important organs, such as the brain, lungs, and liver. Cancer cells continually break off from the primary tumor. Some of these cells may move into nearby blood vessels and lymph vessels. Cancer cells can travel along these vessel highways to other parts of the body. Many cancer cells die while traveling through the blood and lymph vessels, but some may survive and stick to the vessel wall, then move through the vessel wall into another body tissue. These cells may then divide and form a metastatic tumor. Where are cancer cells most likely to spread? Cancer cells tend to spread to certain places in the body depending on the site where the tumor first formed. For example, breast cancer tends to spread to the bones, lungs, liver, and brain, and colon cancer tends to spread to the lungs and liver. It's important to remember that a metastatic tumor is the same type of cancer as the primary tumor. In this example, colon cancer cells have traveled through blood vessels and formed metastatic tumors in the liver. These metastatic tumors are colon cancer, not liver cancer. Metastatic colon cancer is usually treated with drugs and other therapies used to treat colon cancer. Researchers at the National Cancer Institute and other research organizations are studying metastasis and ways to prevent it. Preventing metastasis will help prevent many cancer deaths.
Based on the video, we could see how crucial it is to find a new therapeutic approach in preventing metastatic tumors. We found that these C26 drugs are able to prevent the growth of metastatic tumor. These are our group members, and this is the table of contents on what.
Before that, let's go to the introduction first. What is cancer? Cancer is a disease in which some of the body cells grow uncontrollably and spread to other parts of the body. Normally, human cells grow and multiply through a process called cell division to form new cells as the body needs them. When cells grow old or become damaged, they die and new cells take their place. Sometimes, this orderly process breaks down and abnormal or damaged cells grow and may form tumors, which are lumps of tissue. Tumors can be cancerous or not cancerous, which are known as benign tumors. When removed, benign tumors usually don't grow back, whereas cancerous tumors sometimes do. Hence, the problem here is that many cancer patients relapse often years or decades after their initial treatment and develop new tumors that regrow in the same location or metastasize to other parts of the body. These secondary tumors are often resistant to treatment and are produced by individual tumor cells that may remain dormant for long periods before being reactivated to start proliferating again. This can happen because the treatment didn't destroy all the cancer cells. So, what is C26 drugs? C26 drugs is a new therapeutic approach that will prevent the growth of metastatic tumor. It is a new treatment to prevent the recurrence or spread of various cancer types, including breast cancer and head and neck squamous cell carcinoma, which is known as HNSCC. How it can be happen? The C26 drug forces the cancer cells into a long-lived state of dormancy that characterized by a unique pattern of gene activity. Here are the details how it works. The C26 drugs can activate the NR2F1 protein and hence boosting the level of it. In the previous study, the scientist has discovered that the ability of cancer cells to remain dormant is controlled by a protein called NR2F1. This receptor protein can activate or repress transcription of affected gene by entering the cell nucleus and turn numerous genes on or off, which can prevent the cancer cells from proliferating. NR2F1 levels are usually low in primary tumors but are elevated in dormant disseminated cancer cells. Levels of the NR2F1 protein then decline once more when cancer cells start proliferating again and form recurrent or metastatic tumors. In 19th century, Paul Ehrlich developed the use of chemicals to treat cancer that now we call as chemotherapy. Next, in 2015, the scientist has discovered the ability of cancer cells to remain dormant is controlled by a protein called NR2F1, and after that, in 2020, scientists identified a drug named C26 that can activate NR2F1 by using a computer-based screening, and in the last year, they tested the efficacy of C26 drug on mice as medical in vivo. The mice are injected with patient-derived HNSCC cells, typically form large primary tumors that spread to the lungs after the original tumor is surgically removed. The treatment with C26 drugs should reduce the size of primary tumors, and after surgery, further doses of C26 completely block the growth of metastatic tumors. Instead, the rodent's lungs contain just a few dormant disseminated cancer cells that are unable to proliferate even after the cessation of the treatment.
Cancer treatments has been revolutionized over the years from conventional therapies such as radiotherapy and chemotherapy to a combination of conventional therapies and new developed therapy. However, in many cases of cancer, a targeted approach in improving patient is lacking as every patient is unique. Therefore, each patient cancer treatment should also be specialized. Conventional therapies for cancer such as chemotherapy and radiotherapy remain a primary treatment in cancer. One of the best examples of new technology in cancer treatment is chemo-ID. Small samples of patient's tumor is taken and then used to produce tumor cells in bulk. The cells are being tested with a variety of chemotherapy drugs in order to determine which drugs kill the cell most effectively. Moving on to radiotherapy. There are three types of radiation therapy I would like to discuss today. The first one is intensity modulated radiation therapy, IMRT. This therapy is a computer-controlled linear accelerator that delivers high dose of radiation to a patient's tumor in precisely controlled volumes and size. Next, image-guided radiation therapy, IMGT, which also use linear accelerator, use the technology that can identify the size and shape of tumor within the body, enabling for even more precise radiation treatment. This is especially useful as certain tumors can move and change position as a result of normal bodily function. Another new technology of cancer treatment is high-dose brachytherapy. This therapy utilizes catheter to guide radiation to cancer cells, giving tumors a direct dose. Researchers always find a way in transforming cancer treatment that could bring major breakthrough and make a meaningful difference in patients' life. Therefore, several types of cancer treatment have been established and employed to cancer patients, including immunotherapy, hormone therapy, and targeted therapy. Immunotherapy is a type of cancer treatment that harnesses the body immune system to prevent, control, and eradicate cancer. For example, cancer therapy vaccines can help immune system to detect and respond to these antigens, allowing cancer cells that contain them to be destroyed. Immune checkpoint inhibitors function by preventing checkpoint proteins from forming binding interactions with their partner proteins. Next, tumor-infecting virus is modified virus that used to infect and destroy the cancer that have already formed. Hormone therapy. Hormone therapy is a cancer treatment that delay and stop the growth of cancer that use hormone to proliferate and survive. Last but not least, targeted therapy. Targeted therapy is another type of cancer treatment that regulate cancer cell growth, proliferation, and dissemination. Targeted therapy consists of monoclonal antibodies and small molecules drugs. Monoclonal antibodies bind to specific sites on malignant cells, labeling them so the immune system may easily recognize and eliminate them. On the other hand, small molecule drugs used for targets that are inside cells because they are small enough to enter cells easily. We consider this new drug C26, which is NR2F1 receptor, to be under targeted therapy as it targets the NR2F1 receptor protein to be upregulated in cancer cells, leading to inductions of dormancy to cancer cells. As a result, any cancer that remains after surgery, chemotherapy, or radiotherapy is prevented from spreading to other parts of the body. More detail of this drug will be explained later in the next slide.
Next, mechanism of action. Now, we know the NR2F1 protein is a master regulator of tumor cell dormancy which activate or suppress transcription of effector gene. Level of NR2F1 protein is low in primary tumor but are elevated in dormant disseminated cancer cell, then decline once more when cancer cells start proliferating again and form recurrent or metastatic tumor. So, let we see mechanism of NR2F1 protein. When upregulated, NR2F1 induce expression of dormancy gene signature include the transcription factor SOS9 and retinal acid receptor beta RAR beta. This factor activate expression of CDK inhibitor P27 and P60, which lead to genal G1 cell cycle arrest and cell quiescence. So, how C26 drugs works? The C26 drug act as an NR2F1 agonist. C26 interact with mostly hydrophobic residue in the binding pocket in NR2F1. C26 specifically activate NR2F1, leading to increasing NR2F1 transcription and nuclear protein accumulation. The effect of C26 on the growth arrest was lost when NR2F1 was knocked out by CRISPR-Cas9. The decline of NR2F1 will causing cancer cells start proliferate and form recurrent and metastatic tumor.
Now then, let us move on to the concept of these C26 drugs. According to Khalil et al., C26 is a medication that activates NR2F1 and was discovered using a computer-based screening strategy. C26 was studied in mice to see whether it might prevent metastasis. Then, the animal was injected with HNSCC cells, which are derived from patients who develop large primary tumors that spread to the lungs when the original tumor is surgically removed. The mice were then treated with C26, and it specifically targets and binds to NR2F1 as it acts as an NR2F1 agonist. C26 treatment reduced the size of primary tumors, and following surgery, additional doses of C26 totally stopped metastatic tumor growth. Instead, the lungs of the rodent contained only a few dormant, disseminated cancer cells that were unable to proliferate even when the treatment was stopped. Overall, it is a mechanism-based and rationally designed strategy to exploit NR2F1 activated dormancy as a therapeutic option to prevent metastatic relapse.
Okay, now we proceed to how C26 drug overcome problems in the current cancer treatment. The common problem for the current cancer treatment is it not a targeting treatment or therapy. Means that the treatment is involving all the cells, either normal cells or cancer cells, in that particular area of treatment. But it differs to C26 drug therapy, where it only involving the NR2F1 protein, which act in agonistic way. C26 drug doesn't disturb any cells except the one that has NR2F1 protein, which is involved in the metastatic tumor growth, since it is the NR2F1 agonist. The second problem is an immeasurable drug effect. Common cancer therapy with drug has an uncontrollable effect to other cells, but C26 drug only give it effect based on the intended target. There are studies stated that C26 effect become annual in the NR2F1 knockout cell, which indicate that its effect is only dependent on the presence of the NR2F1. The third problem is a long-term treatment. C26 drug is a self-sustained program which it has a long-lived agonistic effect. Means that C26 has the ability to stop the growth of the metastatic tumor for a long period since its effect is strong enough as a growth suppressive signal. There are also studies mentioned that the effect of the C26 did suppress the growth of THAP3 cell for 1 week after pre-treatment with the C26 in vivo CAM.
Next, we're going to discuss on the latest application of the C26 drugs. In treating patients with metastatic tumor, C26 drug is still in its preclinical study where animals are used as subject in research to observe if a drug, technique or treatment is likely to be beneficial and have significant effect on the subject. There is still no application of this drug on human. However, based on the study done by Khalil et al. 2021, the C26 drug did have positive impact when applied on the mice in the preclinical study. Researchers investigated that C26 could inhibit metastasis in mice. To further describe about the detailed procedures of the study, the mice was first injected subcutaneously with patient-derived HNSCC cells in the interscapular region. This is to induce the tumor cells in the mice. Tumors were allowed to grow and reach a certain volume. The mice are then treated with C26 drugs where it is administered intraperitoneally for 5 days at a dose of 0.5 mg per day. Then, they observe between the mice with control group which is not treated with C26. This was the observation recorded in the journal between control and treatment group. The arrows showed the presence of metastatic tumors in the lung. We could see that with C26 therapy in the treatment group, the number of metastatic tumors in the lungs of mice injected with HSNCC cells was reduced compared to the control group. These results showed that the C26 drugs did have significant effect in preventing metastatic tumors by triggering activation of NR2F1 and causing the tumor cells to remain dormant at the primary tumor sites. This suggests that C26 might probably in the future becoming successful drugs in treating human patients with metastatic tumors.
Based on previous studies of C26 drugs, there are some limitations that need to overcome for better understanding. Firstly, the research have done only limited to head and neck squamous cell carcinoma or certain types of cancer. Second, there is no study that shows how long the cells will remain dormant after treatment with C26 drugs. Third, the toxicity level of these drugs have not yet discovered in this research. For conclusion, by using conventional therapies, some cancer patients find it difficult to recover because cancer can relapse and develop new resistant tumors. C26 drugs is a selective agonist for NR2F1 protein that functions in repressing tumor growth, decrease cancer metastasis, and increase expression of the anti-cancer gene. Since these drugs only able to inhibit metastasis of cancer, therefore C26 drugs is recommended to be used together with another drugs that can induce cell death. C26 drugs also able to increase prognosis of cancer and giving hope to end-stage cancer patients to continue their life. However, C26 drugs still in preclinical study and need further investigation before it can be used by humans safely. Here are the list of references for our product. That's all from our group. Thank you for your attention.